Unit 3, page 3, Enzymes, Factor Affecting Function. Do now, looking at the diagram, explain how an enzyme alters reactions. You can see in the diagram here that for any chemical reaction, it requires some sort of free energy to activate the reaction. Now, let's look. There's a difference when we see without the enzyme, there's a lot of energy required for a reaction to reach the products. However, with an enzyme, what happens to that amount of energy needed for the reaction? That's right, it decreases. So we can see that with an enzyme, it actually reduces the amount of energy needed for a reaction to occur. How do enzymes work? Enzymes work by weakening bonds, which are going to then lower what we just referred to as the activation energy. So again, in this diagram, we see the total energy on our y-axis, and we have the enzyme catalyzed reaction pathway on the x. And we'll see here that in order for the reactant to turn into the product, there needs to be some sort of activation energy, and the enzyme again will lower this activation energy. What are some factors that affect enzyme activity? Well, temperature is number one. A high temperature can speed up enzyme activity, but if it's too hot, enzymes, being proteins, are easily destroyed or denatured. Similarly, with pH, enzymes work best at a specific pH, whether it be an acid, like in your stomach, neutral, like in your blood, or basic, like in your intestines. At the wrong pH, an enzyme can also be destroyed or denatured. The amount of substrate or substance the enzyme is working on will also affect the enzyme activity. If you have more ingredients, you can make more product. There are some environmental conditions that are also going to affect enzymes, like extreme temperatures are the most dangerous. So high temperatures, again, may denature or unfold the structure of the enzyme so that it will no longer have its shape of the active site. The same thing with pH and even ionic concentration. So examining the three graphs, we can see in graph A, we'll see temperature. What's happening to enzyme activity as temperature increases? As temperature increases, enzyme activity increases until we reach about 37 degrees Celsius. What happens after 37 degrees Celsius? Enzyme activity quickly decreases. This is because the enzyme is being denatured or unfolded so that it no longer has that same shape of the active site. What happens with enzyme activity and pH? As pH reaches towards neutral, which is 7, we see that this particular enzyme works very well at a pH of 7, but does not work very well at an acidic pH and does not work very well at a basic pH. And then in the last graph, we see substrate concentration. What happens to enzyme activity as you add more substrate? As you add more substrate, enzyme activity increases, but only until a certain point, because each enzyme can only work so fast. So even if you add more ingredients, it can still only make a certain amount of product at a certain speed. Other important parts of enzymes include cofactors or coenzymes. Inorganic substances like zinc and iron, and even vitamins, are very, very important for enzyme activity. Hemoglobin, which is a blood protein, does not work well without having iron. So iron must be present in the structure of hemoglobin in order for it to pick up oxygen effectively. In summary, what would happen if you overheated an enzyme? It would be denatured and no longer work. What would happen if you took an enzyme from your stomach, a low pH, and placed it in your small intestine, a high pH? It would no longer work because the enzymes only work at specific pHs.